Imagine a condition so debilitating that even a simple breath of air can bring excruciating pain. This is the reality for millions of people around the world living with sickle cell anemia. This inherited blood disorder affects red blood cells, which carry oxygen throughout the body. In sickle cell anemia, these cells become deformed, taking on a rigid, sickle-like shape. These misshapen cells clump together, obstructing blood flow and causing a cascade of health problems. The hallmark symptom is the agonizing sickle cell crisis, an episode of intense pain that can strike suddenly and last for days. Over time, reduced blood flow can damage vital organs, leading to complications such as stroke, acute chest syndrome, and even premature death. The life expectancy of individuals with sickle cell anemia is significantly reduced, highlighting the urgent need for effective treatments. Until recently, treatment options have been limited, mainly focusing on managing symptoms and preventing complications. Blood transfusions, pain medications, and hydroxyurea have been the mainstay of treatment for decades. However, these approaches often come with significant side effects and do not address the underlying genetic cause of the disease. Recent advancements in gene therapy offer new hope for a potential cure. Sickle cell disease affects millions worldwide, particularly in sub-Saharan Africa. Over 300,000 babies are born with sickle cell disease in sub-Saharan Africa each year. The burden extends beyond the individual, impacting families, communities, and healthcare systems. Chronic pain, frequent hospitalizations, and reduced life expectancy take a significant toll on patients and their loved ones. The economic impact is substantial, with high healthcare costs and lost productivity. Despite its prevalence, sickle cell disease has historically received limited attention and research funding. The lack of investment has resulted in a scarcity of effective treatments. However, recent years have witnessed a surge in research and development efforts. Emerging technologies like gene therapy and gene editing offer new hope. The tide is turning, bringing renewed focus to this debilitating condition. Breakthroughs in medicine often emerge from a deep understanding of the disease process. Sickle cell anemia has long posed a formidable challenge to researchers and clinicians. The advent of gene therapy has ignited hope for millions affected by this disorder. Gene therapy aims to correct genetic defects at their root. This technique involves introducing a functional copy of the faulty gene into the patient's blood-forming stem cells. The modified stem cells can then produce healthy red blood cells, effectively reversing the disease's effects. This approach holds the promise of potentially curing sickle cell anemia. Recent advancements have led to the development and approval of groundbreaking treatments. These advancements mark a turning point, offering renewed hope for countless individuals and families. The FDA has approved two groundbreaking gene therapies, Casgevy and Lyfgenia. These therapies represent a paradigm shift in treating sickle cell disease. Casgevy, developed by Vertex Pharmaceuticals and CRISPR Therapeutics, is the first CRISPR-based gene therapy approved for any human disease. This therapy modifies a patient's hematopoietic stem cells, responsible for producing blood cells. Using CRISPR technology, scientists can precisely edit the faulty gene responsible for sickle cell anemia. The modified stem cells produce functional hemoglobin, the protein that carries oxygen in red blood cells. After modification, the patient's corrected stem cells are infused back into their bloodstream. Lyfgenia, developed by Bluebird Bio, delivers a functional copy of a modified form of the beta-globin gene into the patient's stem cells. This modified gene produces hemoglobin resistant to sickling, preventing the formation of sickle-shaped red blood cells. These therapies offer the potential for long-term relief and improved quality of life. They represent a significant advancement in the fight against sickle cell disease. The approval of Casgevy and Lyfgenia marks a pivotal moment in treatment. The promise of gene therapy offers a new lease on life. The approval of Casgevy and Lyfgenia marks a pivotal moment in the fight against sickle cell disease. These therapies hold the promise of transforming lives by addressing the root cause of the disease. Clinical trials have shown remarkable results, with many patients experiencing a significant reduction in painful crises and improved quality of life. For some, these therapies mean freedom from the constant fear and uncertainty of a sickle cell diagnosis. Imagine a life where children can attend school without painful episodes, and adults can pursue their dreams without limitations. These are the possibilities that gene therapies like Casgevy and Lyfgenia bring to fruition. While the initial results are promising, ongoing monitoring and research are crucial. The potential of these groundbreaking treatments continues to unfold. Navigating the frontiers of medicine presents unique challenges and considerations. One significant hurdle is the complexity and cost of gene therapies. These treatments involve a highly specialized and individualized process. This complexity translates into a high price tag, making them inaccessible to many patients. Addressing affordability and access is paramount to ensuring these therapies benefit all in need. Long-term monitoring of patients who receive gene therapies is crucial. Early data suggest lasting benefits, but tracking progress over time is essential. Ethical considerations also come into play with gene editing technologies like CRISPR. The ability to alter the human genome raises profound questions about unintended consequences. Open dialogue among scientists, ethicists, policymakers, and the public is essential. Responsible innovation is key to navigating these complex issues. Unpacking CRISPR technology, a revolutionary tool. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is a groundbreaking gene editing tool. This innovation has opened up new possibilities in the treatment of genetic disorders, including sickle cell disease. CRISPR technology works by harnessing a natural defense mechanism found in bacteria. These microorganisms use CRISPR to defend against viral invaders by cutting their DNA at specific sites. Scientists have adapted this system to target and modify the DNA of other organisms, including humans. By designing a guide RNA to match the sequence of the faulty gene, CRISPR can be directed to cut the DNA at the desired location. Once the DNA is cut, the cell's natural repair mechanisms kick in. Researchers can exploit these repair processes to introduce changes to the genetic code. For sickle cell disease, this means correcting the mutation in the hemoglobin gene that causes red blood cells to sickle. By editing this gene, CRISPR has the potential to produce healthy red blood cells that function normally. The precision and adaptability of CRISPR make it a powerful tool in the fight against genetic diseases. It allows for targeted interventions that were previously unimaginable. CRISPR is at the forefront of research and development efforts aimed at curing sickle cell disease. This technology offers hope for conditions long considered incurable. CRISPR and sickle cell. The science behind the solution. The application of CRISPR technology to sickle cell disease represents a significant scientific breakthrough. At the heart of this approach is the goal of reversing the genetic mutation that causes the disease. In patients with sickle cell anemia, a single nucleotide mutation leads to the production of abnormal hemoglobin. CRISPR-based therapies, such as Casgevy, use gene editing to address this mutation. The process begins with the extraction of hematopoietic stem cells from the patient. These stem cells are then exposed to CRISPR technology in the laboratory. The gene editing machinery makes precise cuts in the DNA to correct the faulty gene. The corrected stem cells are then infused back into the patient's bloodstream. One key target for CRISPR in sickle cell therapy is the BCL11A gene, which suppresses the production of fetal hemoglobin. By disrupting the BCL11A gene, CRISPR can increase levels of fetal hemoglobin, alleviating symptoms. Clinical trials have shown increased levels of fetal hemoglobin and a significant reduction in sickle cell crises. This approach targets the underlying genetic cause, offering a potential cure. CRISPR-based therapies represent a promising advancement in treating sickle cell disease. Real-world impact stories from the front lines. The true measure of any medical breakthrough is its impact on patients' lives. Victoria Gray's journey with CRISPR therapy for sickle cell disease shows its transformative potential. After treatment, she experienced fewer pain episodes and a newfound sense of normalcy. Daniel's health improved significantly, allowing him to enjoy activities like sports and school. These stories underscore CRISPR's potential to change lives. Voxelotor, a new weapon in the arsenal. While gene therapies have taken center stage, another treatment option has emerged: Voxelotor. Voxelotor targets the root cause of sickle cell disease by preventing red blood cells from contorting into their characteristic sickle shape. This medication represents a significant advancement because it addresses a fundamental aspect of the disease process. Unlike traditional treatments, voxelotor tackles the underlying mechanism responsible for the sickling of red blood cells. This approach has the potential to alleviate symptoms and prevent long-term damage. The development of voxelotor highlights the importance of understanding the molecular mechanisms driving disease progression. By targeting specific protein interactions, voxelotor offers a more precise way to combat sickle cell disease. The availability of voxelotor expands treatment options, providing an alternative for those who may not be eligible for gene therapies. This is particularly important for patients in regions with limited healthcare resources or those facing challenges related to the cost and complexity of gene therapy. Understanding voxelotor mechanism of action. Voxelotor binds to hemoglobin S, preventing clumping and sickling of red blood cells. This improves blood flow, reducing sickle cell crises and complications. Enhanced oxygen delivery alleviates symptoms and prevents organ damage. Voxelotor exemplifies the power of targeted therapies in modern medicine. Voxelotor on the NHS. The NHS in England has approved voxelotor for individuals aged 12 and older. This decision expands access to innovative treatments for sickle cell disease. Voxelotor addresses the underlying cause of sickle cell disease. It significantly improves quality of life and long-term health outcomes. This inclusion is a testament to collaborative efforts in advancing sickle cell research. The availability of voxelotor is a beacon of hope for sickle cell patients in England. Triumph over adversity, stories of hope and healing. The true measure of any medical breakthrough lies in its ability to transform lives. In the realm of sickle cell disease, stories of hope and healing are emerging. These narratives offer a glimpse into the life-altering impact of innovative treatments. Consider the inspiring journey of Aisha. Diagnosed with sickle cell anemia at birth, Aisha's childhood was punctuated by pain crises. The fear of these episodes loomed over her life. However, Aisha's life took a dramatic turn with a CRISPR-based gene therapy. The treatment was a resounding success. Aisha's pain crises subsided dramatically. She could envision a future free from sickle cell disease. Her story is a testament to the transformative power of gene therapy. A new dawn for sickle cell patients. David noticed a remarkable improvement in his health. His pain crises decreased, and his energy levels soared. He returned to playing basketball, a sport he loved. David's story underscores the importance of innovative treatments. Aisha and David's experiences represent a new dawn for sickle cell patients. Section 3. Beyond the Horizon, the future of sickle cell treatment. The horizon is bright with promise. Researchers and clinicians are tirelessly working to refine existing therapies and explore new frontiers. Gene editing technologies like CRISPR offer the potential for more targeted interventions. Scientists are exploring new CRISPR-based approaches that could permanently correct the sickle cell mutation. The future of sickle cell treatment is bright with hope, fueled by scientific advancements and collaborative efforts. Section 1. A new era of hope. Breakthroughs in sickle cell anemia treatment mark a pivotal moment in medical history. For decades, families faced a future clouded by uncertainty and pain. Gene therapies like Casgevy and Lyfgenia, and medications like voxelotor offer a beacon of light, transforming treatment and offering hope. These advancements represent a paradigm shift in treating this genetic disorder. By targeting the faulty hemoglobin gene, they offer potential cures. Stories of transformed lives remind us of the human impact of scientific progress. These advancements improve quality of life and empower individuals. They represent hope for communities disproportionately affected by this disorder, promising a future where sickle cell disease no longer dictates lives. Section 2. The power of innovation and collaboration. The progress in sickle cell treatment shows the power of human ingenuity and collaboration. Gene therapies and CRISPR have transformed the field. This journey involved countless hours of research and clinical trials. Collaboration has driven innovation and accelerated progress. Equitable access to these therapies is crucial for a future free from sickle cell disease. Ensuring access requires collaboration among all stakeholders. Section 3. Towards a future free from sickle cell disease. Advancements in sickle cell treatment offer a glimpse into a future where this disease no longer dictates lives. Research and new treatments brighten the outlook for those affected. The horizon is ripe with possibilities, fueled by gene editing, stem cell therapies, and novel drugs. Promising avenues include refining CRISPR for precision and efficacy. Scientists aim to permanently correct the sickle cell mutation in stem cells. Techniques like base and prime editing expand the genetic toolkit. Novel drug therapies target different aspects of sickle cell pathophysiology. These include increasing fetal hemoglobin, reducing inflammation, and improving blood flow. Stem cell transplantation is also being explored as a curative option. The journey requires commitment to research, innovation, and collaboration. Effective, accessible, and affordable solutions are essential. Progress shows what can be achieved when united in a shared mission. I am sure if you have come this far, you have learned a lot, so please subscribe for those new and healthy lifestyle and share. Let us work together to make sickle cell disease a relic of the past.